Welcome, class. It's Dr. Lindner. In this video and the next few relating to neurophysiology, we're going to be talking about, you're going to be hearing words like, let me see if I can write some of these down for you so that you're going to be hearing things like action potential. You're going to hear things like graded potential. You're going to hear terms like, we're going to go over sodium, potassium pump. We're going to be going over things like resting membrane potential, the RMP, resting membrane potential. We're going to be talking about terms like threshold. We'll be talking about depolarization. We'll talk about repolarization. So there's depolarization, repolarization. We'll also mention hyperpolarization. So depolarization, repolarization, hyperpolarization. What else will we mention? You're going to hear of numbers like minus 70 millivolts, minus 55 millivolts, positive 30 millivolts. These are terms that you're going to be hearing. And yeah, I think that's a pretty good start. We'll also be talking about, you'll hear words like EPSP and IPSP, which stands for excitatory postsynaptic potential and inhibitory postsynaptic potential. And then we'll be learning about, you'll hear neurotransmitters. So lots of neat things and terms that are going to be coming up. So we're going to start with the neurons and electrical signals in neurons. So let's review and understand that in terms of neurology, we remember that there were different types of cells that made up the neural system. There was the neuron and there were neuroglial cells. And the neuron is the only one that is electrically excitable, and it's electrically excitable due to a voltage difference across their membrane. And we'll see if we can understand what we mean by a voltage difference across their membrane. We'll talk about the different types of electrical signals that are delivered through neurons. One of them is an action potential and another is a graded potential. So let's take a look at this short little clip, this short video here.
Simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the neuron. This is a brain. Estimates vary, but right now the best guess seems to be that our brains contain around 85 billion neurons. The neuron is a nerve cell, and it's the primary functional unit of the nervous system. This is a generic image of a neuron. Neurons actually come in all shapes and sizes, but this is the prototypical version of a neuron that you'll often see in a textbook. The structures extending from the left side of the neuron that look a little bit like tree branches are called dendrites. Dendrites are the area where neurons receive most of their information. There are receptors on dendrites that are designed to pick up signals from other neurons that come in the form of chemicals called neurotransmitters. Those signals picked up by dendrites cause electrical changes in a neuron that are interpreted in an area called the soma, or the cell body. The soma contains the nucleus, which contains the DNA, or genetic material of the cell. The soma takes all the information from the dendrites and puts it together in an area called the axon hillock. If the signal coming from the dendrites is strong enough, then a signal is sent to the next part of the neuron, which is called the axon. At this point, the signal is called an action potential. The action potential travels down the axon, which is covered with myelin, an insulatory material that helps to prevent the signal from degrading. The last step for the action potential is the axon terminals, also known as synaptic buttons. When the signal reaches the axon terminals, it can cause the release of neurotransmitter. These purple structures represent the dendrites of another neuron. When a neurotransmitter is released from axon terminals, it interacts with receptors on the dendrites of the next neuron, and then the process repeats with the next neuron.
So that's a little bit of a review of the neuron. Now, within the neuron, in terms of electrical conduction and action potentials that you heard.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I would.
Right. So in order for those action potentials to be transmitted or produced, we need to have an action potential that's being generated. In order to do that, there's a lot of things that happen in relationship to what's called a resting membrane potential, the RMP. So within the cell membrane of the neuron, we have different types of channels. Remember, there's proteins on those cell membranes, and one of the functions of proteins is they can act as channels. Some of those channels are called leakage channels. Some of them are called gated channels. A gated channel sounds as it says. There's a gate or a door that's there, and it can open and close in response to different types of stimulus that can result in the excitability of the neuron. One of those gated channels is called a voltage gated channel. And a voltage gated channel means that when the voltage or the membrane potential starts to change a little bit, it can open and close. Ligand gated, when you see that word ligand, we are going to think that they're going to open and close in response to some sort of chemical stimuli. And maybe the chemical stimuli is a hormone, maybe it's a neurotransmitter, or maybe it's an ion. And gates can open and close based on mechanical stimuli like vibration or pressure. Okay, so those are gated channels. Leakage channels are as they sound. There's no gates. They're always open. It's almost like one of those old school hoses where some people have like in-ground sprinklers and some people have a hose that has a sprinkler on it. And some people have a long hose that has those perforated holes throughout. And what happens when you turn on the water is that water leaks out of those perforated holes of the hose. Okay. The nerve cells are going to have more potassium leakage channels than sodium leakage channels. So the nerve cell is going to have more channels to leach potassium than it does sodium. And as a result of that, the permeability to potassium is much higher. And what ends up happening when potassium is leaking out more than sodium can rush in, it creates this unequal type of membrane potential of what's outside of a cell compared to what's inside of a cell. And the outside of a cell membrane of a neuron tends to be a little bit more positive in relationship to the inside of the wall of the cell membrane that tends to be a little bit more negative. And the RMP, the resting membrane potential of a neuron, is minus 70 millivolts. That's the resting membrane, meaning if this is a chart, and let's say we have minus 70 here, and then we have minus 55, and then maybe a 0, and then positive 30. When we're over here along the minus 70 millivolts, this is at rest. If you start to see something like this, then there's some sort of activity or some sort of action potential that's being generated. Otherwise, it's at minus 70. That is resting membrane potential. Everything's at rest. So these are examples of different types of channels where what we're looking at here, this should look familiar as a phospholipid bilayer, right? This is the cell membrane where you've got your phosphate, your hydrophilic portion here, your other hydrophilic portion here, and the hydrophobic portion here, right? These are the phosphates, and then the lipids are on the insides. So this is a voltage-gated channel, and it's closed. We see the doorway right here is closed, so nothing can make it through to the other side. Okay, and that is RMP, a resting membrane potential of minus 70 millivolts. But something is happening where we can see that these little pyramids here, these little triangular things look like they're potassium, and there's 1, 2, there's 3 of them there, and those 3 are giving it a minus 70 millivolt charge. But if one of the 3 moves into the channel, we become less negative, right? When you go from minus 70 to minus 50, we have less negativity. We're moving more positive. So when that happens, that stimulus of the change in the voltage is enough to open up that gate so that potassium can exit. Now, below it is a chemical stimuli, where again, you have your phospholipid bilayer. Here's a channel, and the channel is closed. But it looks like we have some sort of chemical. When you see that red triangle, that's usually indicative of acetylcholine. So now we have acetylcholine, that's the ligand, right? So it's a ligand gated channel. When the acetylcholine binds, now we start to have things moving in both directions. So that's another example of a gated channel. And then the third type is the mechanically gated channel, where there's some sort of vibration, some sort of pressure, some sort of mechanical stimulus. And we can see here that here it's closed on the left-hand side. Then there's some mechanical stimuli which opens up the gate, and now we start to get the entrance of certain ions that are passing through. Okay, the dark blue is typically outside of the cell, so this is extracellular. And then whenever you see the tan, that's usually the cytosol, which is intracellular. So let's take a look at what happens. I did mention that there's a little bit more of a negative ions along the inside of the cell membrane and a little bit more positive. So if this is the cell membrane, a little bit more positive on the outside and a little bit more negative on the inside. And that's just due to the difference of what we have out here, which is more sodium and maybe chloride. And what we have more on the inside is more potassium. And because of this difference between sodium and chloride and potassium, on the inside, we also have amino acids. That's what gives this a little bit more of a negative charge. And the sodium, a little bit more of a positive charge on the outside. Okay, so the difference between the ions and cations and anions between outside and inside is going to give us this resting membrane potential of minus 70. All right. So what gives it that minus 70? It's the concentration of the ions that are different of what we have inside the cell compared to outside. So extracellularly, the fluid is rich in more sodium and chloride, and the cytosol inside is more full with potassium, phosphate, and amino acids. Okay, and the membrane permeability, remember, it's different for sodium and potassium because the membrane itself is more permeable to potassium. So you have potassium that's has a higher concentration inside than outside, and based on concentration gradient, potassium wants to flow outside and sodium wants to go inside. But the inward flow of sodium, it can't keep up with the outward flow of potassium due to the permeability. And then we have what's called that sodium potassium pump that tries to remove the sodium that leaked in, that gets pumped in, it tries to push it out. So let's look at this sodium potassium pump and how it works, okay? See if we can get that to open.
If you want to travel with your kid,
Sorry about the commercial, it'll
you
be
don't
over
want
in
to
a
share
second,
your
yeah.
kid's bedtime. You might not want to share a hotel room.
Okay.
The sodium potassium pump is a protein pump found in the cell membrane of all animal cells. Its main function is to transport sodium ions out of the cell and potassium ions into the cell. This serves a number of purposes, but is critically important to the function of neurons as it helps to maintain a higher concentration of sodium ions outside the cell and a higher concentration of potassium ions inside the cell.
Okay, that's what the sodium potassium pump is trying to do because in its state, there's a higher concentration of sodium outside and a higher potassium inside. But potassium goes from intracellular to extracellular, and sodium goes from extracellular to intracellular. But to get it back to where it began, you're going against its concentration gradient. Whenever you're moving something against the concentration gradient, that's where you need ATP or energy.
Preserving these differences in ion concentrations helps to stabilize the cell's membrane potential. This is critical for the neuron to be able to fire an action potential, which is the basis of the electrical signaling of neurons. The sodium potassium pump is a large membrane-associated enzyme that uses the energy of adenosine triphosphate, or ATP, to transport sodium and potassium ions across the cell membrane. All of the details of how this is achieved are not yet fully clear, but the general process involves the binding of ATP to the pump, which promotes the binding of 3 sodium ions and the release of 2 previously bound potassium ions. ATP is broken down and transfers a chemical group known as a phosphate group to the pump, which prompts the pump to undergo a conformational change, or a change in its shape. This causes the bound sodium ions to be released into the extracellular fluid. At the same time, the pump binds 2 potassium ions, which also prompts another conformational change that returns the pump to its previous configuration, and the cycle begins again. Each cycle of the pump causes 3 sodium ions to move out of the cell and 2 potassium ions to move into the cell. Because there is one more positive charge leaving the cell than entering, there is a net loss of positive ions, which makes the resting membrane potential of the cell slightly more negative.
Give me a second. I want to see why this didn't open up very much. Let's see if I can figure out why that was. Okay, let me rewind that a little bit so we could see that again.
Across the cell membrane. All of the details of how this is achieved are not yet fully clear, but the general process involves the binding of ATP to the pump, which promotes the binding of 3 sodium ions and the release of 2 previously bound potassium ions. ATP is broken down and transfers a chemical group known as a phosphate group to the pump, which prompts the pump to undergo a conformational change, or a change in its shape. This causes the bound sodium ions to be released into the extracellular fluid. At the same time, the pump binds 2 potassium ions, which also prompts another conformational change that returns the pump to its previous configuration, and the cycle begins again. Each cycle of the pump causes 3 sodium ions to move out of the cell and 2 potassium ions to move into the cell. Because there is one more positive charge leaving the cell than entering, there is a net loss of positive ions, which makes the resting membrane potential of the cell slightly more negative.
Okay, so they're saying that you see you've got these 3 sodiums and then you got 2 potassiums. So as these 3 sodiums are pumped to outside of the cell, whereas only 2 potassiums are pumped in, there's more of a net positive charge outside of the cell, and there's more of a negative charge in relationship to that on the inside of the cell. Great. I'm glad I was able to open that up so everyone could see that. So in the resting membrane potential, there's a voltage difference. And I just kind of use this visual to kind of show how you've got extracellular fluid, you have intracellular fluid, here's your cell membrane, and there's positives and negatives outside and some positives and negatives inside. But the net difference creates a little bit more of a positive charge outside extracellularly and more of a negative charge intracellularly. And that voltage difference creates the resting membrane potential. When they put that voltmeter inside and they measure it, it gives us a resting membrane potential of minus 70 millivolts. Now here we can see on the left-hand side right here, let me change this. So again, we see extracellular fluid. Here's the cytosol, intracellular. You can see that there's more of a positive charge outside of the cell, more of a negative charge inside the cell. We can see that outside of the cell, you've got some sodium, you've got chloride. On the inside of the cell, we've got more phosphates. You have more Na, more sodiums out here, and you've got more potassiums in here. And there's also protein, right? So all of that, when you look at the net charge, creates a little bit more of a negative inside and more positive outside. You can also see that when you have potassium going out and you've got sodium coming in, you don't need any ATP for that. You don't need any energy. Why? Because if there's more sodium outside, then the concentration gradient says things go from a higher concentration to a lesser. So it's going to move intracellularly. And because there's more potassium outside than inside than outside, then potassium is going to go from an area of higher concentration to less. But once that happens, we want to try and restore it back to balance. And in order to do that, that's where this sodium potassium pump kicks in, where it takes energy to pump out 3 sodiums and to pump in 2 potassiums. And that voltage difference creates more of a positive charge outside and negative in relationship to that. Okay. Okay, let's take a look at this short video on an action potential. Let me pause this and see if we have any questions so far.